Hi everyone and Assalamualaikum. Today we are going to learn about electricity generation from animal dung using microbial fuel cells technology. Let's get started. People throughout history have used manure to burn for fires when no trees or wood could be found. In 10th century, farmers around the world have obtained energy from their livestock manure. But in 1911, M.C. Porter, a professor of botany at University of Durham, have the idea of obtaining energy from bacteria. In his studies of how microorganisms degrade organic compounds, he discovered that electrical energy was also produced. Porter had the idea of trying to harvest this newfound source of energy for human use, and he was able to construct a primitive microbial fuel cell. After almost 100 years, researchers have fully understood the microfuel cells and how to generate electric from it. What is microbial fuel cell? Microbial fuel cell is a process to generate electricity directly from manure or organic matters by using bacteria as catalysts. Microbial fuel cell acts as bioreactor by using bacteria to convert chemical energy stored in biomass to electricity. Microbial fuel cell consists of anaerobic anodic zone and aerobic cathodic zone. Bacteria attach at the surface of the anode to pull electrons out from the organic matters. The electrons travel from the anode and flow to the cathode to produce electrical current. Let's move on to the bacteria characteristics. Microorganisms used in microbial fuel cell will act as anaerobic digester to produce biogas from manure to generate electricity. Usually, Bacillus species is used in microbial fuel cell as it have the ability to transfer electrons and is also classified as electrochemically active bacteria or electrogenic bacteria. Bacillus species is a gram-positive bacteria and have the root-shaped structure. Others electrogenic bacteria which are Shawiniella putrescens and Geobacter sulfuridescens also can be used in microbial fuel cells to generate electricity.
Concept of MFC to generate electric. There are 6 basic components of MFC: cathode, anode, anodic chamber, cathodic chamber, proton exchange chamber, and electrode catalyst. Elements of microbial fuel cell. A MFC is a bioelectrochemical system that drives an electric current by using bacteria. Typically, MFC apparatus consists of 2 chambers, anodic and cathodic. These 2 chambers are separated by a proton exchange membrane. The anodic chamber contains organic substrate such as cow dung. Cathode chamber contains a high potential electron acceptor to complete circuit. The MFC chambers can be constructed by glass, polycarbonate as well as plexiglass. Materials such as carbon cloth, carbon paper, graphite and graphite felt can be used in anode electrode, while cathode can be made up of material such as platinum or platinum black catalyst material. Major element of microbial fuel cell: proton exchange membrane, anode chamber under anaerobic conditions, cathode chamber under anaerobic conditions, substrate to feed on, pure or mixed bacterial culture, biofilm, nitrogen gas, anode electrode, copper wire, resistor, cathode electrode, and air oxygen. Why cow dung can be used in MFC technology? Because it is efficient, renewable resource, and also good sources. Characteristic of dung. MFC fed with cow dung showed that with a higher moisture content was more suitable for current generation. Based on one of researchers state that high moisture content resulted in the maximum power densities in generation of electric.
Safety requirement in MFC. First parameter is the preparation of materials. It is in order to achieve better result as it determines the amount of output produced and its performance. Wear protective gear during handling MFC system, and special room are required to keep animal dung. This is because the waste from animal dung does have high amount of nitrate, which able to give harmful effects to human health. Products that produced in the entire MFC system. Other products besides electricity are: first, hydrogen that can be used and harvested into biohydrogen production. Second, carbon dioxide produced in the anode chamber. And third, we have methane that produced from animal waste. Chemical reaction involved in electricity production from animal dung. MFC microbial fuel cell consists of anode chamber, cathode chamber, and separated by a proton exchange membrane (PEM). In anode chamber, active biocatalysts from animal dung will oxidize the carbon source or organic substances and generate electron and proton. Then, proton in anode chamber are conducted through the PEM to the cathode chamber, while electron produced are conveyed through the external circuit. Electrons will reduce the electron accepted in the cathode chamber when they pass through. While electron and proton being transferred, the parallel oxygen reduction to water occurs. Next, in cathode chamber, protons and electrons produced in the anode chamber complete the electrical circuit. Protons and electrons will react to form water molecules in order to be able to spread via a permeable membrane in the cathode with the aid of catalysts. The performance of cathode and yield in cathode depending on the electron acceptor, the concentration and species of the oxidant, the performance of catalyst, and the structure and availability of the electron.
What makes animal dung produce a maximum voltage? First, the concentration of distilled water and animal dung. Equal concentration of cow dung and distilled water helps in the diffusion of electrons and protons easily, so the oxidation and reduction process can take place efficiently. The types of water. Mixture of cow dung slurry with untreated domestic water exhibit a very high microbial population. This what happens when you mix the cow dung with water. Abundance of microbes will utilize the organic compound in a biowaste to produce electricity. Third, the type of microorganisms. It seems that cow dung slurry and untreated domestic waste has both gram-positive and gram-negative bacteria. And then we have the anodophile and cathodophile microbes. The anodophile microbes can be found in the anodic chamber, while cathodophile microbes can be found in the cathode chamber. And then we have the electrogenic microorganisms, which act as mediator to transfer electrons to the anode indirectly. Fourth, microbial activity. The roles of microbes are very important because they help to oxidize the carbon compound of biowaste in anodic chamber to produce electrons and protons. They even help to form biofilm around the anode so that the electrons can be transferred directly to the anodes instead of electrogenic microbes indirectly. When to replace the extracted animal dung with new fresh dung. They should be replaced when there are no equal concentrations of animal dung with distilled water, and then when there are high density of microbes, they must be replaced because they will bring nutrient exhaust. This can be seen when Pseudomonas and Bacillus species are cultured together. The differences between fresh dung and extracted animal dung. Fresh dung has higher organic carbon content. It has lower electrical conductivity, it has higher minerals and nutrients, it has lower antimicrobial activity, and finally, It releases harmful gases such as methane gases.